Introduction. Welcome to today's lecture on medical microbiology focusing on gram-negative bacteria. In this comprehensive lecture, we will explore the various aspects related to the structure, characteristics, clinical significance, mechanisms of infection, antibiotic resistance, biofilm formation, pathogenesis of specific infections, endotoxins, diagnostic strategies, host immune responses, and novel therapeutic approaches associated with gram-negative bacterial infections. Gram-negative bacteria possess distinct features that contribute to their pathogenicity. One such feature is the presence of an outer membrane, which acts as a permeability barrier and protects the bacteria from harmful substances. Additionally, gram-negative bacteria have a unique cell wall composition, consisting of a thin peptidoglycan layer surrounded by an outer membrane. These structural characteristics are crucial in understanding the mechanisms of infection and developing effective treatment strategies. Gram-negative infections are of great medical importance due to their prevalence and impact on patient outcomes. These infections pose significant challenges in terms of treatment, often requiring potent antibiotics and prolonged therapy. Students will gain an understanding of the clinical significance of gram-negative infections, including their association with healthcare-associated infections, community-acquired infections, and their impact on vulnerable populations. Gram-negative bacteria employ diverse strategies to cause infection. These include adhesion to host tissues, invasion into host cells, and the production of toxins that damage host cells. Students will explore the specific mechanisms utilized by gram-negative bacteria to establish infection and evade host immune responses. Understanding these mechanisms is crucial for the development of targeted therapeutic interventions. The ability of gram-negative bacteria to acquire resistance to antibiotics presents a significant challenge in clinical practice. Students will delve into the mechanisms through which gram-negative bacteria develop resistance, including the production of enzymes that degrade or modify antibiotics. This knowledge is crucial for the rational use of antibiotics and the development of innovative strategies to combat drug resistance. Efflux pumps play a key role in multi-drug resistance exhibited by gram-negative bacteria. These pumps actively pump out antibiotics from the bacterial cell, reducing their efficacy. Students will explore the molecular mechanisms underlying efflux pump-mediated resistance and its implications for therapeutic interventions. Horizontal gene transfer is a significant contributor to the spread of antibiotic resistance genes among gram-negative bacteria. Students will gain insights into the different mechanisms of horizontal gene transfer, such as conjugation, transformation, and transduction. Understanding these mechanisms is critical for developing strategies to limit the dissemination of antibiotic resistance. Biofilm formation is a hallmark of many chronic gram-negative infections, making them challenging to treat. Students will learn about the complex process of biofilm formation, the role of extracellular polymeric substances, and the associated resistance to antimicrobial agents. This knowledge will help in developing novel therapeutic approaches to combat biofilm-related infections. This topic will focus on specific gram-negative bacteria, such as Pseudomonas aeruginosa and Escherichia coli, and their associated diseases. Students will gain an in-depth understanding of the specific mechanisms and virulence factors employed by these bacteria to cause infections, enabling targeted therapeutic interventions. Endotoxins, particularly lipopolysaccharides, play a crucial role in the development of sepsis and systemic inflammatory responses associated with gram-negative infections. Students will explore the molecular basis of endotoxin-induced immune activation, the cascade of inflammatory events, and potential targets for therapeutic intervention. Accurate and timely diagnosis of gram-negative bacterial infections is essential for effective patient management. This topic will introduce various diagnostic techniques, including molecular methods such as polymerase chain reaction, PCR, and nucleic acid sequencing, as well as conventional culture-based methods. Students will gain insight into the strengths and limitations of each diagnostic approach. Gram-negative bacteria have evolved sophisticated mechanisms to evade and manipulate host immune responses. Students will explore the interplay between host immune mechanisms and bacterial immune evasion strategies, enabling a comprehensive understanding of the host-pathogen interaction. Lastly, this topic will focus on emerging therapeutic approaches for tackling gram-negative infections. Students will explore alternative antimicrobial agents, such as bacteriophages, immunotherapy, and antimicrobial peptides. These novel approaches show promise in overcoming antibiotic resistance and improving treatment outcomes. In conclusion, studying gram-negative bacterial infections is crucial in medicine. Understanding their characteristics, infection mechanisms, antibiotic resistance, and interactions with the immune system is vital for effective treatments. Developing advanced diagnostics and therapies can improve patient outcomes in combating gram-negative infections. Thank you, and I hope you found this lecture informative.